Hi there, and welcome to our transcriptomics sections. So, first of all, we are going to discuss what is transcriptomics. It's the comprehensive study of RNA transcripts that may include mRNA, ribosomal RNA, transfer RNA, and other coding RNAs produced by the genome at a specific development stage or underlying particular physiological conditions. So, what is a transcriptome? Transcriptome refers to complete set of RNA transcript that is expressed in a cell, tissue, or organism at a given time. It includes all types of RNA such as messenger RNA, ribosomal RNA, transfer RNA, and non-coding RNA which are microRNAs, long non-coding RNAs, etc. So, transcriptome is dynamic and can be vary depending on the cell type, developmental stage, physiological condition, or disease state. Understanding the transcriptome is crucial for studying gene expression, regulatory mechanism, and very essential for identifying potential biomarkers or drug targets. What are the techniques for this is the RNA-Seq, which is high throughput sequencing of entire transcriptome, which allows for the comprehensive analysis of gene expression levels, alternative splicing, and identification of novel transcripts. Then there is hybridization technique, which is based on expression of thousands of genes in parallel and very limited, less sensitive RNA-seq. Then we have PCR, which is highly sensitive and accurate method for quantifying the expression of specific gene, often used to validate the findings from high throughput technique. So the data we find through RNA-seq and microarray, the genes that we found out, we validate them by using the PCR. So what is RNA-seq? RNA-seq is the powerful technique that uses next generation sequencing to profile entire transcriptome of a cell or a tissue. It involves extracting RNA, converting it to cDNA, and fragmenting the cDNA and then sequencing the fragments to obtain billions of short reads. These reads are then computationally aligned to the reference genome, allowing the quantification of gene expression level, and that leads to the identification of novel transcript, alternative splicing event, and RNA modification. So all of these steps we are going to perform in our practical session. RNA-seq offers several advantages over traditional microarray technology for transcriptome. It offers high resolution and sensitivity to detect low abundance transcript. It has ability to identify novel transcript and splice variant that are not represented on microarray chips. So it also provides the quantitative measurement of gene expression level across wider dynamic range. And it also requires less input material and has low background noise. Then there is experimental design. You can also do it experimentally. You have to choose your biological sample. After that, you have to extract it RNA. After extracting the RNA, you have to prepare library of it. After preparing the library, we will send the samples to a specific company. You can send them to a specific company like Illumina, and then they will make a transcriptome for you and they will analyze. After getting the data, you have to be sure how to analyze the data. So that is the computational part. So we have raw data processing that consists of millions of short reads. These reads must be pre-processed to remove low quality adapters. So that we are going to do. After that, we have alignment of reference genome and then quantifications of gene. After quantification, we then lead to the normalization of data, and then we have some quality control checkups as well along the way. Differential gene expression analysis is identifying differentially expressed genes, which genes are significantly low expressed, which are very highly expressed like this. Then we have fold change calculations as well, in which ratio the expression levels of compared conditions were changed. Then we have p-value, that p-value is the significance of differential expression. And then we have discovery rate correlation as well. Also, we can differentially express genes are visualized using heat maps, volcano plots, and other graphical representation to interpret the results as well. Then there are applications of transcriptome. It is used in biological research for the gene expression pattern and fundamental biological processes. Then we have precision medicine. We can use them to identify biomarkers of diseases that we are going to do in microarray section. And then we also use them in biotechnology areas as well. Then they are also used in transcriptomic studies that shed light to the evolution of gene and gene expression across species. Then there is advancement in this. There is single cell RNA-Seq that is now being used in advance. Then we have long read sequencing that improve the transcriptome assembly and isoform formation. Then we have multimodal omics. The different data omics are being integrated into transcriptomics as well. So transcriptomics evolve, and by using this we have a deeper understanding of cell to cell heterogeneity. Additionally, the integration of transcriptome with other data such as proteomics or epigenomics will provide a more comprehensive view if we take this in the future. So that is just a little bit introduction of transcriptomics and RNA-seq.